Welcome, everybody. Happy Friday. We are back with another Frontiers in Neuroscience, in which we cover a lot of the most exciting topics, the most pressing topics, and relevant, of course, to brain health. I'm Dan Krozik, the Deputy Director here at the Center, and we are a cognitive neuroscience center devoted to neuroplasticity. We also have an online audience, so if you're chiming in from afar, go ahead and use the Q&A function to ask questions as they come up. We will have time for questions at the end of the presentation today. So neuroplasticity is the topic of the day, and what a speaker to have our own Dr. Mike Kilgard from the UT Dallas campus. Now, Mike did his undergrad out at UC Berkeley. He then stayed on the West Coast for his PhD at UCSF, working with none other than Mike Merzenich, who is a legend in the neuroplasticity community, and Mike was working on the auditory system at that time. He then moved to UT Dallas in the late 90s and joined the faculty here, where he has done some of the most careful neuroplasticity work targeting the vagus nerve and evoking neuroplastic responses largely within animal models, and did that for, and continues to do that work, but gradually moved toward human subjects. So he has done just tremendous work in neuroplasticity. Mike is now the Interim Executive Director and Chief Science Officer at the Texas Biomedical Device Center, in addition to holding the Margaret Fond Johnson Professorship at UT Dallas. He's offered over 100 peer-reviewed articles. He's run 9 clinical trials looking at tinnitus and PTSD and stroke recovery. And additionally, Mike has been tremendous at fundraising for his work and really driving this work forward. He's had a major role in DARPA funding. DARPA crafted their TNT and SPARK programs largely around work that Mike had done the foundations on. So really tremendous scholar, and I learned a lot of new things every time I hear Mike talk. So we're really excited to welcome Mike Kilgard.
Thanks, Dan. It's always good to be here at the Center for Brain Health. I'm going to be talking today about topics I presented at Walter Reed Army Hospital a few months ago, and then the American Spinal Injury Association. So we're going to be sort of merging these two ideas. And at Walter Reed, I began with this slide that I thought was helpful for them to think about what progress in brain health is really going to look like. And it's similar to the progress we've made in automobiles, where it's taken us 100 years to move from the basic idea of 4 wheels on the road to something that really works well today. Incremental progress year after year after year has led to a remarkable improvement in the quality of cars. Similar progress has been made in the immune system. It took about 100 years for the Army to be able to develop a treatment that's effective for malaria. And it required really deep understanding of the immune system and how it operated to be able to regulate it appropriately. It took about 100 different failures before we were able to have what we have today, which is a truly effective therapy for malaria. This has happened time and time again over the past 100 years. The immune system presented ourselves with problems that were impossible, and we eventually eliminated them one by one. For us, of course, who work on the brain, that's not where we're at today. We're not at a time where we're reducing 99% of brain impairments, and we're trying to reduce a few percent. And that's because there's so many different problems. Developmental problems, acquired problems, problems with mental health. And we've made a lot of progress understanding the underlying biology with support from the National Institute of Health. We've spent about $100 billion on neuroscience research, and we have an enormous amount, about 3 million papers have been published on the brain. But I have to report, of course, that there's some challenges. We thought it might be simple. This is a classic paper from some years ago. We've hunted for some big, simple neuropathological explanations for psychiatric disorders and have not found them. We've hunted for big, simple neurochemical explanations for psychiatric disorders and not found them. We have hunted for big, simple genetic explanations for genetic disorders and not found them. So we thought, well, let's do some imaging and see if we can understand the human brain better with imaging technology. And a more recent paper, despite 3 decades of intense neuroimaging research, we still lack a neurobiological account for any psychiatric condition. So this seems really daunting. But this is very similar to where we were at the beginning of our understanding of the immune system and infectious disease. So we have to understand the relationship between cause and effect, and it can often be quite complicated. In the case of COVID, we had an enormous success. There's a simple thing causing a serious problem, and we know the relationship between the complex symptoms of COVID and the initial underlying biology of this infectious particle. So it's probably better for us to think about things that are a little more complicated than something like COVID and infectious disease. So cancer and autoimmunity are a little bit more in the realm of complexity for psychiatric disorders, and in many ways neurological disorders as well. So if you think about the tens of thousands of different genes that contribute to the functioning of the cell, you understand a little bit better why cancer is not something that we snap our fingers, make a vaccine for, and make go away, but we make incremental progress day on, day on. For the brain, we certainly can understand the brain circuits. This is the basal ganglia circuit. And we all know well that in Parkinson's disease, certain neurons perish, and we can do interventions that are really inspired by understanding the underlying circuits of the brain. Many have heard of L-DOPA or deep brain stimulation that are effective because we understand how the brain works and what that circuitry, how that circuitry leads to pathology and eventually recovery. Now, the complex circuitry in the brain is hard to overestimate. It's 100 billion neurons. So even if you just put a few areas, I don't think this really captures the complexity. Thinking about how could it be that 100 billion neurons see and have conversations and all that is still something we struggle with. So I like this artist's rendering of just this rat's nest of connectivity. It's 1,000 trillion synapses, and you got to figure out which ones to strengthen and which ones to weaken to help people get better and recover from their injuries or problems with mental health. So I began my career in this much simpler organism. This is Synergitis elegans, a simple free-living animal that lives in the soil all around the world. It's got 302 neurons, and we know every single neuron, its exact date of birth, who it came from, what it connects to. We now know the connections between every single synapse in every single cell in this animal. And we've known this for actually some decades. But again, the idea that if we knew the circuit, we could explain the behavior isn't really true. Behavior cannot be explained even when the entire genome and the entire connectome has been known for decades. And that's because behavior is an emergent property of these connections. But progress in cancer and rheumatology makes it clear that we don't have to understand everything. We don't need perfection. We need good enough to make a difference in someone's life. So what we need is precision targeting. My background is in biochemistry, and it seemed like we would need a drug that could target a specific location. I'm happy to say that we have lots of drugs that target lots of very specific areas, but they tend not to be very effective. I'm going to talk about post-traumatic stress disorder, and it's not clear that there's a really strong effect of targeting some particular molecule in the brain to treat PTSD. So the idea from the immune history is if we could target the connections, the native plasticity inside of the immune system, we get vaccines. And if we could target the native plasticity of the nervous system, we might be able to direct new synaptic connections to help people recover from serious neurological and psychiatric disorders. So this began with work in the 1970s. Hugo Wiesel got the Nobel Prize for figuring out how the eyes worked, how they hook together, and how they make images in the brain. And fast forward pretty quickly, these were doctors trying to understand how can I help someone who has a childhood condition called amblyopia. And they developed this idea that you should do reverse patching. After you remove the occlusion to one eye, you need to block the perfectly good eye, which is kind of counterintuitive, but it leads to incredible outcomes for these participants. By rewiring their brain, there's nothing wrong with their eye, by rewiring their brain, they get an extra line on the normal eye test you see. And these benefits are long-lasting. Once you produce the right plasticity, these kids get to keep these gains for a long time. Fast forward to the 2020s, and now we have VR goggles and fun games to play. It's a little bit less onerous than walking around with a patch all day long. So we can make technology make these kids' lives a little easier. So the question is, what else could we do where we could take the ability of neurons to make new connections and direct them in an appropriate way to help someone in the clinic? So how are we able to drive enough of the right kind of synaptic plasticity to improve mental health? One of the most successful therapies is quite surprising. It's still electroconvulsive therapy for major depressive disorder, where having a seizure produces a large amount of neuroplasticity, and that plasticity in this very severe condition improves outcome and quality of life for these people. We'd like to be a little bit more targeted than a generalized seizure, and so we're working on more precise technologies. Much of the progress has occurred in the psychology domain, cognitive science domain, and so this is from the VA website. The technology of using cognitive behavioral therapy has improved year after year as we do very detailed, double-blind, placebo-controlled trials to really understand how do people respond best, what changes are needed in the brain. So that things you have to do, it has to be systematic, deliberate, repeated, prolonged, and unconditional if you're a therapist. And what that means is it's got to be gradual, not too fast, not too slow. We don't want to push. It's got to be deliberate. It's got to be under the patient's control. It has to be repeated. You can't do it once in a while. People with PTSD are exposed to their triggers all the time, but you need to do it at just the right interval. Not too fast, not too slow. You can't do it for a minute. You got to do it for a long time. And it needs to be unconditional. They can't believe that there is some other safety cue, rabbit's foot or something else attributing to it. And with this incredible technology, we've been able to eliminate PTSD in about 40% of people who undergo this therapy. About 85% of people respond, get better. But you can view that as a glass half full or glass half empty. Half of them still have PTSD after receiving this very challenging task of revisiting each of their fears over and over and over for many, many days in a very difficult way. Among our military veterans, the success rate is about half. So about 20% would have loss of diagnosis, which means 80% will retain the scars they suffered. So we're interested in trying to understand how better to optimize synaptic plasticity to improve mental health and neurological diseases. And so this takes us back quite some time as we thought about how does the brain operate? What are the key parameters that are regulating whether or not the brain changes? And we know that when you see a stressor, it could be something that reminds you of an improvised explosive device or sexual trauma, or in this case, a king cobra. And there's, of course, actions that go into the brain that are very arousing and distressing that lead to changes in the body. But there are also changes in the body that then in turn distress your brain as you realize my heart is pounding and I'm sweating. And we've been interested in attacking both of these pathways simultaneously. So Dr. Krausik mentioned the idea of using vagus nerve stimulation, and the rationale here is we can't ignore the body, we can't only attack the body, we have to treat them both simultaneously. And after dozens and dozens of failed approaches that didn't work at all, mostly pharmacotherapies, but also deep brain stimulation and others, we settled upon vagus nerve stimulation because it offered a number of very unique opportunities that you couldn't get anyplace else. One is it had very good temporal precision. When you take a drug, the drug is there every second for many, many, many minutes, or probably hours. Whereas learning occurs on a second-by-second basis. You get a tennis serve, something happened at that moment, and we can activate the vagus nerve precisely in time using electricity to this nerve. We also, though, want to relax the body, and the parasympathetic system, as you may have heard, activates the parasympathetic system, vagus nerve, and that's the rest and digest system. So we're arousing the brain while we're relaxing the body, and this is the optimal conditions for learning. And we had a prediction that it might lead to therapeutic relief for PTSD if you were to combine vagus nerve stimulation with this prolonged imaginal exposure therapy. So again, there's lots of nerves. They all come out of the base of the brain. One we're focusing on here is the 10th cranial nerve, the longest cranial nerve. And the key things to know is that there's an excellent safety profile. Most of the time we have good ideas. It doesn't work because it isn't safe. It causes side effects that are off target. That's a real problem. Most of these fibers are sensory fibers. The brain is learning about the body mostly through the vagus nerve. And so the way it knows when the heart and lungs are beating and pumping appropriately is because of information coming up from the viscera. These fibers we're interested in are very easy to activate. They're very large diameter, and so tiny amounts of current activate the handful of fibers that we want to activate, which are the stretch receptors from the heart and lungs. And when we activate them, we've shown in animals and in humans, you get a very brief, like half a second burst of the key neuromodulators that regulate synaptic plasticity, acetylcholine and norepinephrine. And the key is that most people don't even notice it. So although you were sending this powerful signal, most people go about their business, wouldn't even know this is happening. So in terms of safety data, there's about 70, almost 80,000 people now who've received a lot of vagus nerve stimulation. They get 30-second trains every 5 minutes, 24 hours a day, 7 days a week for decades. And this reduces their epilepsy by about a factor of 2. And so that told us that the tiny amount of stimulation we were planning on presenting was likely to be safe. And to skip to the punchline, it is safe. We did a series of experiments, beginning with a Nature paper in 2011, and now it's almost 60 papers on this topic, in animals trying to tease apart, is it possible to use a brief burst of vagus nerve stimulation to drive plasticity in the brain? And as Dr. Krausik mentioned, we started with tones, but then we went with Ben Porter through the use of movement. We could also drive changes in the motor system. And then we began a series of experiments in animals with stroke or spinal cord injury or other conditions, demonstrating that we could improve recovery even in severe injury models. I'm proud to say we were able to translate that with our clinical colleagues into human trials, first in tinnitus with Sven Van Est and others, and then more recently with Jesse Dawson, an Auser engineer, into a series of stroke studies that culminated in a design where we're doing typical physical therapy. You have trouble moving your hand, let's practice moving it, do challenging tasks over and over. These people would be implanted with this vagus nerve stimulation device. And I'm proud to say that that led to a series of successful clinical trials, ending in a Lancet study, first authored by my colleague Jesse Dawson from University of Glasgow. And then in summer 2021, the Food and Drug Administration approved this as the first treatment for people with stroke. So the first brain injury during the chronic phase treatment that's ever been developed. All developed here at UTD initially from animal studies, making it all the way through to FDA approval. Now, I'll get back to the motor aspect in a little bit, but thinking about if we can drive change in the motor system, could you also drive changes that might help with mental health, in this case, post-traumatic stress disorder? So first challenge is, you really want a big giant battery implanted in your body and a connector if you've just got PTSD. Well, people with PTSD don't think of it as just having PTSD, but Rob Renneker at the Texas Biomedical Device Center in the School of Behavioral and Brain Sciences at UT Dallas thought he could make a device that's a little bit better than the 1980s technology that you see in a pulse generator sitting under your collarbone. So he shrunk the device down by about a factor of 60. So that device is about the size of your pinky nail, and it sits now directly on the vagus nerve. So there's no wire, single incision instead of two, minimally invasive, takes about 30 minutes to put in. So that's the device. It's tiny. FDA declared it a breakthrough. We've now implanted 35 people here in the Dallas area with this new technology. So I want to tell you about those 9 individuals who've received this implant who have chronic post-traumatic stress disorder that was not responsive to other previous therapies. So the idea is each person will undergo implantation, then they will undergo the standard, the gold standard of care, prolonged imaginal exposure therapy, and the therapist will have this new tool. It's just a cell phone, and this cell phone's got a little button on it. When you push that button via Bluetooth, it activates the external device that's been worn around the neck, and that goes through the skin and activates the device that's implanted and sits directly on the vagus nerve. This we predicted and supported with animal research done with Krista McIntyre and colleagues up on main campus. It suggested that we would reduce anxiety. It's really important for this exposure therapy to work for people to be anxious. So if you take a benzodiazepine, this therapy has no utility whatsoever. This, however, is a way where we're enhancing the neuroplasticity in the brain. Benzodiazepines, of course, suppress learning because they suppress anxiety. And vagus nerve stimulation has this unique combination of relaxing the body by activating the parasympathetic system while it enhances neuroplasticity by the central release of the neurotransmitters I mentioned before. So we're going to do this imaginal therapy, recording it on the phone. We may as well use the phone as a recording device, so they're actually recording their own traumatic event, retelling in first person. And then they go home and listen to it again and again and again. So you can imagine someone who survived one of these traumas hearing it again and again and again. We want them to say what we ended up hearing them say, which is, I'm so bored of hearing this story. And so they go home and listen to the same story retold now in their own voice again and again and again, each time putting this device back on, and now they're receiving vagus nerve stimulation at home, precisely timed to the delivery of this therapy. So here's the trial design. Done with Mark Powers and Marie Warren and a number of others at the Center for Trauma Research at Baylor University Medical Center, just a few miles from here. We inform consent, implantation, 12 office visits, and then an assessment, and we follow them up for 6 months after. So first off, many people were worried, well, you're not going to get people to mental health to have an implant. They all said, sign me up for an implant. We recruited at exactly the same rate for this trial as we'd recruited for all the previous trials that didn't have an implant. Which is about 1 per month. Every person we asked at the end said they would receive this implant again if they were given a choice, and they would recommend it to others. So what are the results of this study? It's not yet published. We're working on getting there. We have finished our 9th person and we ran out of money, so I think we will probably end there. But vagus nerve stimulation is definitely safe. It is definitely well tolerated, even during such a stressful activity as being reminded of the most difficult day of your life. And we've shown that it reduces the clinician-administered PTSD score for the DSM-5 and 2 other measures, standard assessments for PTSD progress. So this is a comparison. This is an open-label study, so we do not have a placebo control, but we do have historical controls. So the white bars are several hundred people who received conventional prolonged imaginal exposure therapy, And they began at roughly the same point, about 40 on the CAPS-5 score. After therapy, they drop. What we found was that we increased the amount of drop, and the benefits appear to be quite long-lasting. In the 5 individuals we had data for at the time of making of this preliminary analysis, all of them were no longer meeting the diagnostic criteria, so we expected 40%. We're about somewhere between 80 and 100% so far in this trial, no longer have PTSD. We, of course, need to do this study right. We're seeking funding from the National Institute of Mental Health, having some challenges there, but the hope is we can really prove this out by doing a double-blind, placebo-controlled trial. Everyone has the device, but it's only activated in half. And then if there are some who fail to benefit from the therapy, maintain their diagnosis of PTSD, we would then cross them over and they would repeat the therapy now with the device being engaged, so that every person would have an opportunity for remission of their symptoms. So just to summarize this piece from my talk at Walter Reed, the idea is amblyopia proved the concept that you could use targeted plasticity if you knew what to do, in this case reverse patching. Stroke could be physical therapy, and now imaginal therapy for PTSD suggests that you need to drive the right type of changes in the right places. The problem is that these responses are often partial. We do not typically receive complete recovery, and we think the reason for that is we're not producing sufficient amounts of neuroplasticity, and we think we can do better by adding vagus nerve stimulation. So here's just the same therapies, adding vagus nerve stimulation, and we see greater recovery. That's our goal. So just to take you back a little bit as we move forward, what are the next steps? This is now FDA-approved treatment, so I want to talk about kind of next steps. Much of this goes back to our animal research, which we never shut down. So this is an animal reaching out, had a stroke, is trying to grab that bar and pull. When they deliver the appropriate amount of force, we can deliver a pellet down, and the animals do this task over and over and over again. And this is what led to the FDA approval. This is Seth Hayes' work and Naveed Kodapras's work when he was a graduate student in my laboratory. And what we were able to do is take animals, train them on this task, they know how to reach and grab this thing and pull. Then we give them a stroke, and they end up being a lot weaker, as you might not be surprised. So their hit rate goes from 90% success down to 40%. Then we train them every single day for all their food, and they get better. But if you were to have added vagus nerve stimulation, those who got randomized to that group make, in fact, a complete recovery, and that recovery lasts well after the end of vagus nerve stimulation. So that was the rationale, but what's the basic science? How is this working? Why is vagus nerve stimulation operating? Is this really magic, or is there some underlying biological understanding? And so if you don't understand the biology, you really shouldn't be delivering the therapy. And so we were able to use a technology developed at NIH that allows you to inject particular muscles in the forelimb. Those muscles then are infected and transmit the viral particles all the way up into the brain, transsynaptically labeling and then expressing fluorescent proteins that literally glow like Christmas trees, and you just go in and count the cells. These are the cells in the brain that are connected to that particular muscle. And what we can see is that rehab is good. You do increase the number of neural connections when you do lots of therapy, but you do not produce enough neuroplasticity to make a complete recovery. You could do better with the remaining tissue. And we can see here we're producing about 4 times more connections in the brain if you have the exact same therapy adding this extra burst of neuromodulator. And one simple way to think of it is that physical therapy is pretty boring. Even prolonged exposure therapy, if you do it day after day after day after day, I've done this before, it's probably not like a life or death experience. But when we activate the vagus nerve, certain regions of the brain think that you're having a heart attack, and they're releasing a lot of neurotransmitters, and they don't adapt. Every time they do it, you're not conscious of it, but the arousal network of your brain is sending something that's saying, we should make some changes, and we should make them now. So we first began with stroke. We did lots of animal models as a meta-analysis, consistently saying we could improve outcomes in rats, whether they were young or old, male or female, all the rest. We went on to do a series of studies in other indications that led us to the trials I'll talk about now, which is spinal cord injury. First is just a summary. I've already mentioned this work, this Lancet study on the right, but we've done trial after trial, first open label studies, then double-blind placebo control, and then finally the large-scale multi-site international pivotal trial. That was 108 people. So what's left to be done? Are we done? You do your basic research, then you get to the FDA and you move on. The answer is no. There's a lot of work to be done. The improvement we've made is significant, it's meaningful, clinically meaningful. The number needed to treat is 2, so if you get this therapy, you have a 50-50 chance of making a clinically meaningful difference. But that means there's still disability. It's not like these people who receive this therapy don't have stroke. You would be able to tell. Their hand doesn't work the normal way. But we think we're at the beginning, just like we were at the beginning when Jenner had this idea of having people exposed to cowpox as a prophylactic for smallpox. It's not a very good vaccination, but it's better than nothing. And early vaccine technology got better and better and better with systematic parametric studies. So first thing we've been doing is working on developing a much smaller device, cheaper to make, more rapid to implant. I've already mentioned that to you. What I want to talk about now is the projects we've been doing to maximize the benefit. In that Lancet study, those people made those gains in 18 days. So they had a stroke that removed a significant fraction of their brain, and yet they really only got vagus nerve stimulation 18 times. So what happens if you do more than 18 times? So we have approval at UTD to do 144 sessions. In the people in our trials, this is quite a bit more than 18. Now the problem is you got to do 144 sessions of physical therapy, which is its own challenge. How would our healthcare system be able to deliver 100 therapy sessions? Who would pay for it? How would we do it? How many trips to a major medical center would you have to make? How would you pay for parking and gas and all of that? But the hypothesis was simple. Maybe 18 is as good as you're ever going to get. There's plateau in physical therapy. But maybe with vagus nerve stimulation, you could make additional gains if you did more. Maybe it's linear. Maybe you can make massive gains. We don't know how much better people could get. So with a new device that Rob Renneker built, it's tiny. We're able to activate these things during physical therapy now in people with spinal cord injury and hemorrhagic stroke. We've only done ischemic stroke to date. The key thing we learned in those trials is that the therapy has to be of the highest quality. You can't just take any old movement and impair it. It must be challenging. In the same way, the PTSD therapy has to have all 5 of those criteria. They're very well understood criteria that drive neuroplasticity. Even though it doesn't lead to dramatic gains, we do know how to deliver physical therapy appropriately. So the question is, how are we going to deliver physical therapy at home? And how are we going to know when to activate the vagus nerve? If you're just sitting around watching TV, we don't be activating it then. So we need a closed loop system that can talk to this device in their neck while they're doing physical therapy that's engaging, motivating, arousing, and challenging. So we need to be able to individualize it. You're not going to have one size fits all. It's got to be hard. They got to be working every day making errors, or they're not going to make progress. The clinicians and scientists are going to need to be able to track their progress remotely, presumably over the internet. We're going to need to give them real-time feedback. They need to know, am I succeeding? How am I doing? When do I succeed? When do I fail? And then we're going to need to be able to synchronize the vagus nerve stimulation precisely in time. So first, just the individualization. We do an assessment, either the Fugel-Meyer upper extremity subscore, which is what we use for stroke. We target where someone is still having problems. If they can't move at all, you're not going to be able to train that. If they're normal, you don't need to train that. So we train those tasks where they have, in this case, a 1 on a scale of 0 to 2. For the GRASP assessment for spinal cord injury, it's a 6-point scale from 0 to 5. We're looking for 3s, so we would be targeting those areas where there's room to make progress. It moves a little bit. These are people with incomplete injuries. It's not a transection. They have connections, they just have damage. So we're going to individualize it a lot, but every person's going to be different. Where their scores lie is unique to every person. So we need to be able to deliver a wide range of exercise at home. I have a grant from NIH with our partners at Flint Rehab who developed this puck technology, and we've added a bunch of new technologies at the Texas Biomedical Device Center to allow people to turn knobs and handles to type with one hand, to swipe playing Fruit Ninja, but also to lift their shoulder or do a fly out or practice pinching with their fingers. In order to keep it challenging, we can use normal video game technology. We don't want to get distracted by the video game, so something pretty simple. Ball goes back and forth, you drive a little car, but it allows you to lose track of time as you're moving back and forth. For us, pinching this back and forth as she's doing after her cervical spinal cord injury is easy for us, but it's difficult for her to get the motivation, to get those neurons to fire, and in particular to get her fingers to open. And so she can practice that day after day. Here's another individual using the 2 different ones. This puck has a force sensor, so when you squeeze it, you can detect it. That's what's on the upper right. But also the angle is measured with an accelerometer located inside the puck. And so we can shoot these little fruit and make it challenging. So you have to squeeze what would for them be very hard after a stroke. A paper just came out a few weeks ago from David Pruitt at the Texas Biomedical Device Center, showing that you could gamify all these things. Make it not just repetitions where you just do more, count steps, but actually play some games and make it a little more fun. So that's a list of the different games that interact with these different technologies. You might be typing. We could find words you can type with one hand after a stroke, for example, and then that word is written on a shark, and you type that word, and the shark goes away, that kind of thing. So here's Space Runner, challenging task, person squeezing the task. If they're really weak, how are they going to do the task, right? This task is impossible if you're too weak. So we multiply their force by a factor of 10. so that they can play the game, cross the threshold, and activate the jetpack and the little flying guy so he can go up and down. But we only multiply it by just enough where they can do it, but it's still challenging. So as they get better, you might multiply it by much less as they get stronger. We want to get it just right so they can sometimes do it, but not always. And that sweet spot we can then adjust over time. It may take 100 sessions, it may take 500 sessions, we don't know, to recover from a serious problem like ischemic stroke or hemorrhagic stroke. So as we adjust it, we then write that number down, and that becomes our metric of progress for that patient. So this is one of our patients. Over these 36 sessions for the initial phase in the early feasibility trial, you can see at first we were multiplying the scores by 10, and over time we're reducing it proportionally by about 20% each time we can. If you make it too hard, then they can't do it, and they say I can't play the game, and then you have to go backwards. That then correlates to the improvement in pinch force. The last one was on a log scale. This is a linear scale, but you can see over time he's getting better and better and better and better. And what began as something he could do 20, 30, 40 grams, eventually it's 200, 300, 400 grams. And we see this time after time. People are making progress. As they practice, we keep making it harder. They produce larger and larger forces. But these scales, it's sort of hard to imagine, what do these scales correspond to? So if you're trying to pull a paper clip off, you'd need to be pinching about 120 grams. If you want to cut paper with scissors, you got to go close the scissors at about 256. If you want to close a pair of tweezers, that's a little bit heavier of a spring. You need to be using 500. And one of those binder clips you've probably seen, that's a lot of force. So even though they're making progress, they're still not back to normal. Normal uninjured controls can produce 8,000 grams. So we're making progress, but there's still a long way to go to get rid of the deficits after chronic cervical spinal cord injury or stroke. We want to give the participants real-time feedback. So we give them first an extensive in-clinic instruction. We don't just send them home with these things. They're doing the therapy. They played all these games at home already. They know how they're working. They're experts. We also send them home with videos where a licensed physical therapist is explaining to them how to hold it, what to do, how to handle common problems. What do you do if this happens? So if they get into trouble, you can hit the help button and there's a therapist suddenly pops up and reminds you what we're supposed to be doing, because it's a lot. We have supervised telerehabilitation. Caitlin Malley is one of the students in my lab right now, and she just gets online. Someone opens their thing just like you do for Zoom, like a lot of teachers did during COVID, and they get on and they give the instructions and they help the participant make progress and make changes in an efficient way. And then she can also directly stimulate the vagus nerve. We've been giving them continual visual feedback. Joe Epperson and Emmanuel in my laboratory are PhD students, and they've been really working hard, again with David Pruitt, who graduated some years ago now, to make sure these games are visually appealing, but you can literally see when I'm doing well. If I squeeze harder, I get more of a response, and so that continual visual feedback is really important. And then we can now produce tracking logs and show them how were you doing over time. Your Fitbit does that, your Apple Watch does that. I'd like to be able to do that as well for my physical therapy. And traditional physical therapy doesn't have anything like that to document progress, which is highly rewarding. This is just an example of what one individual, this is a stroke participant, is doing. It's individualized to his particular impairment. He has trouble pronating and supinating his hand, has trouble lifting, has trouble feeding himself. And so we're doing a series of activities that match his particular deficits, all of which I think you can tell are challenging, but they're all doable. Doing something impossible is not going to get them anywhere. Doing something challenging, plus vagus nerve stimulation, we've shown that in a randomized trial, physical therapy alone is good, but physical therapy with vagus nerve stimulation is better. So here's just an example, zooming in on one of these tasks. You're turning, in this case, the knob doesn't actually turn, but we measure the forces. So we're plotting in physical units of torque, it's in Newton meters, how hard are you turning? And this allows us then to use that as a continuous closed loop. If you're turning above average for you, I can detect that. It's 5% better than you usually do, and I'm stimulating only on those trials that are better than average for that person to help direct the neurons to make the right connection changes to move toward greater strength in this case, or range of motion in other cases, or speed. So now we can track someone. This is just one therapy session where someone is doing a series of different tasks. The green dots are showing the 382 times when an automated algorithm reads what the forces are being produced and delivers vagus nerve stimulation, precisely timed a movement of the hand that is better than average for this particular person. So here's just an example of a few of our first spinal cord patients, showing you kind of what they're doing at an activity of daily living. I just need to pick up a key or a coin. That's really hard for them. So we pick coins that are doable but challenging, and so we do a combination of tasks that are computer-based, and also a bunch of tasks that are real-world tasks, picking something up, putting a card in a slot, things like that. So again, here's a different spinal cord individual now doing his particular set of activities that meet his deficits at this time. What you'll see in the background is the physical therapist for yet another participant, and you'll see they're not needing to do that much, which they're really excited about, because their job is very difficult, to be constantly hands-on. And so we're imagining a world where the physical therapists are really writing the prescriptions, and they're supervising what's going on, making sure people aren't cheating, aren't hurting themselves. And that's an excitement that the people, Chad Swank is one of the directors at the Baylor Institute for Rehabilitation, again, just a few miles from here. Rita Hamilton is the director of that program, and they've just been fantastic in embracing this new technology, developed up with computer technology up at UTD, and then putting it into the clinic and seeing how do participants enjoy. How do clinicians who are not normally trained to use these kinds of technologies, how do they embrace them? Well, if you do a good job, they love it. If you do a bad job, they say this is too complicated. I'd rather go back to my stretchy band and my weights. So the feedback from the clinicians is just as important as the feedback from the participants. And uniformly, they think this is really getting them to be fully engaged, staying at their challenge levels for the entire session. Documented, well-published papers have shown most people only do about 20 or 30 repetitions in an hour of physical therapy. And many people who've had physical therapy say, I don't want to go, I didn't do that much. And that's just because you get talking, you get distracted, the games keep you focused, and the ability to turn the knob exactly, precisely. If you can do a 1 pound weight, you can't do a 2 pound weight, what are you supposed to do? Here we can turn it to be 1.8, 1.9, 1 point, whatever you need to be. So this is just a diagram. This is ongoing. As you can see, we started this particular trial in April of 2021, and this is going all the way up until today. So we've now done 21,405 exercises in 25 people. You'll see it's a mix of people with ischemic stroke, hemorrhagic stroke, now for the first time, and cervical spinal cord injury. And we're now beginning to ask this question. How far can we go? What else is possible? Once you had an adjuvant, this used to be aluminum salts before we had mRNA vaccines, we'd put aluminum into the vaccines, and that would irritate the immune system and say, you should come fight this thing, this is a really bad thing. It didn't really matter whether it was measles, mumps, and rubella, or whether it was flu. The same thing could be used to direct plasticity in the immune system for very different disorders. And the hope now is that we have a technology that allows us to activate the vagus nerve, trigger the release of these neurotransmitters. We can label specific events instead of labeling specific pathogens. And it's now up to us to figure out how would we use it. But we really imagine this like the early days. You have a Model T, it kind of drives. If you've ever been in one, it's not so great. It's backfiring a lot. It's not really very fast. It's not very safe. There's no airbags. It's got lots of problems. But we think over time, now that we have this new tool, which is safe and easy to use in individuals, we can make forward progress on a variety of disorders that we really haven't made much progress, despite many billions of dollars spent understanding the brain. And the hope is not only will we be able to help people get better, but we'll also be able to close that loop and understand the underlying biology. What are we getting in these brain readouts? What is the EEG telling us? Once you can change the brain, it's a really powerful tool for being able to see what is causal in the kind of measures we've made at the basic science level as well. So just to sort of wrap up, some conclusions. Neuroscience has produced very few tools that allow us to safely enhance synaptic plasticity in people. You could do the eye patch, you can have the seizures, there's some things like that. You sort of do physical therapy, but they're typically not producing life-changing improvements without serious side effects or lots and lots of time. Vagus nerve stimulation, as I've described, is able to enhance plasticity in both humans and animals, and that plasticity is sufficient to improve recovery. It doesn't mean they recover fully, but they make gains they couldn't otherwise make with practice alone. And the key really is this activation of the parasympathetic system, the triggering of the release of these neurotransmitters. I've mentioned the neuromodulators, acetylcholine, norepinephrine, and serotonin. And so the idea then is we'll take the same principles we learned from infectious disease and cancer. You've got to target it. You've got to help the system know. Most of the work is not being done by us. When you fight off a bad infection, whether it's COVID or strep throat, the drugs aren't doing that much. Your immune system is doing all the real work. But if we can direct the immune system appropriately, you can do a much better job. And the COVID vaccine reduced deaths by a factor of 10. And And that's sort of amazing. That's what success looks like. Not a little better, not 15% less disability, but 90% less disability. That's what we're shooting for. And we now have reason to believe we might be able to get there, but it's not going to be fast. It wasn't fast for the aircraft, it wasn't fast for the automobile, and it's unlikely to be fast here. It'll require lots of sustained research activities supported by both private foundations and federal governments. So we're learning how to direct therapeutic synaptic plasticity, but the prediction is this is going to be hard. So I want to just take a few minutes and thank the people who really made this all go. They're not all on this slide, unfortunately, but most notably Dr. Rob Renneker, who was the founding director of the Texas Biomedical Device Center, and the engineer who produced the device that allows us to run our clinical trials. Seth Hayes was there at the very beginning of the very first animal experiments in stroke, and continues to be instrumental in every single trial we do. We were joined by an incredible clinician in Jane Wigginton at UT Southwestern in the emergency medicine department, who was willing to try some new things. She'd seen all the worst of the worst in emergency medicine and realized there wasn't much to do. She can save their lives, but then they have this ongoing disability. And she jumped in both feet and allowed us to really do something we had never done before, running clinical trials, large numbers of them, here in Dallas. It's really quite a challenge. Institutional review boards, I know you guys all know, was challenging. We needed to make sure that we protected patient interests at every moment. We've been able to do that with her support. Mark Powers is the director of trauma research at Baylor. Scott and White, Baylor University Medical Center, jumped right in and said, I've tried lots of things that didn't work. He tried exercise and a bunch of things that were thought to be useful adjuvants for prolonged exposure therapy and reported that they didn't work, and jumped right into this and said it probably won't work. But if it does work, we'll move forward with it. If it doesn't, we'll try something else. And that's the kind of approach we've taken. Mike Foreman is the head of trauma surgery at Baylor University, Baylor Scott and White. Also said, I'll learn that surgery, I'll put those things in. General surgeon, but was really interested in the application of something new for the treatment of psychiatric and neurological disorders. Rita Hamilton, director of the Baylor Institute for Rehabilitation, and Chad Swank, one of the scientists there, also directing it. Long list of physical therapists, undergraduates, graduate students, and all the rest. Don't have time to thank everyone, but it's really an incredible team and an honor to be the director of the Texas Biomedical Device Center, where all these people work together every day to try to make new treatments for people where there are currently no options. I had a slide before that said questions, but I don't have my question slide anymore. Thanks.
Mike, thank you so much. Really inspiring and tremendously careful work. We do have time for questions, so go ahead and raise a hand if you have one. I'll kick things off by just, I wanted to ask about the specific parasympathetic activity that you spoke about, how that is dampened down by vagal nerve stimulation. And in PTSD, it would seem as if you'd have very, very high peripheral nerve activity and probably extremely high central nervous activity when a extremely debilitating anxiety-provoking memory is formed. So I wonder if you could comment on that interesting difference between the sort of very, very high sympathetic system that's not optimal for memory, yet you get these tremendously strong memories, and then how that works in terms of your model.
Yeah, so it's a great question, and in humans it's really hard to know. I mean, what all is happening, we don't know, but luckily this work was based on dozens of animal experiments done in collaboration with Krista McIntyre and Jimenez D'Souza, Lindsay Noble up on main campus, and so we really do know the answers to these things. In animals, of course, you can stick the electrode and record, and so certainly we do see abnormal activity both in the brain and the periphery. There's really no question about that. The question was, could you intervene and do anything good, or are you just going to make things worse? And the answer was, VNS alone is anxiolytic, so it definitely acts in the way you would think. Animals are less afraid when you activate their vagus nerve because, again, you've activated the rest and digest system. They notice that the heart rate is lowered and their blood pressure is lowered and they're feeling more calm and assured. But you're also simultaneously releasing these neurotransmitters. We weren't sure what would happen if you release these neurotransmitters in the brain while you calm the body. We were certainly worried that by enhancing neuroplasticity, at the time you're talking about the trauma, you might make them worse. That was certainly something we were greatly concerned by. Our first participant was a U.S. Army veteran, and so last thing we want to do is make his nightmares more severe. He doesn't have PTSD anymore, and that was because we believed the animal work. We believed that the animals didn't get worse. And the idea that humans aren't animals, of course that's true. But careful science really should lead to things that translate from animals to humans. And in this case it did, both for stroke and now in an open label study for PTSD. So the mechanisms remain to be fully elucidated, but what we have done is to record the connection in animals between the prefrontal cortex and the amygdala, and we see real powerful changes there that occur not with vagus nerve stimulation alone, but only when you combine the vagus nerve stimulation with the memory of the trauma. So we're not trying to turn a circuit down or up, we're trying to turn that specific memory, that specific ability, and that's where the targeting comes from, is that precise timing. So vagus nerve stimulation alone, not very helpful. For epilepsy, it can reduce seizures by about 2 if you do a lot of it. But we're delivering 500 times less. This is a tiny amount of vagus nerve stimulation, but it's precisely delivered at the moment of experiencing this either motor experience or sensory experience or trauma reminder.
It's always scary when the non-neuroscientist asks a question. How much of this neuroplasticity translates, like for someone with a spinal injury, whether it's cervical, lumbar, whatever, is there neuroplasticity in the nerves themselves, or are you really taking advantage of what's going on in the brain to process that information differently? Because it could change post-surgical treatment and that sort of thing.
That's right. Yeah, so great question. Again, in the humans, well, you can't see, you can't look in the spinal cord to figure out what's happening. In the animals, of course, we can. The animals make the ultimate sacrifice, and so we're able to look and literally count the cells, see where those connections are. So you have to be using incomplete injury. Before we started in humans, just like we tried 10 different models in animals with PTSD, we did 3 different models of spinal cord injury in animals to make sure it really did work, not under one condition, but all the conditions we tried. And so the key is that you have to have some remaining connection. If there's no connection, maybe then if you added stem cells, you could make a connection, and then if you added this, it would work. But if you don't have a connection at all, it would be hopeless for our therapy. So we're thinking about a day where it would eventually need to be combination therapies for the more severe individuals. But for those who have spare pathways, what we see is where you get restored function depends upon where there are pathways that remain. So in some cases, some of the fiber tracts are completely obliterated. The connection to the cortex, for example, the corticospinal tract, is completely gone. How could they get better? Well, there's still a connection to the red nucleus, another brain area in the brainstem. So long as there's a pathway, the brain has a way to figure it out. Except that it doesn't. It didn't figure it out. Those animals didn't get all the way better. But when we add vagus nerve stimulation, giving this extra cue, that allows it to sort through the literally billions of possible synapses. Who should I strengthen? Who should I weaken? I mean, look at all the connections you can imagine just with the handful of neurons above you. What would it be like if there were literally 100 billion? And you had to figure out, I don't want to lose all my memories. I don't want to lose my speech, my language, all this. I just want to change a few. And that's the problem that the immune system has. How do I change my immune system to get ready for COVID? Turn it up? No, that'll cause autoimmune disease. Turn it down? No, that'll cause other problems. I've got to turn it just right. And that's really where our challenge lies. With the immune system, it's done with antibodies and binding and these sorts of things. With the brain, it's really done with circuits. And the timing is what gives us the temporal precision. So when a kid learns to ride a bicycle, they're wobbling around and, oh, I did this, and they're putting it together. Don't do that, don't do that, do this. So we're adding that signal in individuals who can't do it for themselves because they've had a stroke. They've had a spinal cord injury, and so their sense of touch is not good, their evaluative networks are not good, their motivation, they're often depressed. All those systems are impaired. We can provide that back, not permanently, but just during physical therapy, hoping that then we can step back and let them keep the gains they've made.
Has there been any thought of looking at other ancillary improvement in cognitive function? You kind of alluded to that.
Yeah, great question. So Dr. Rittman's asking about cognitive impairments and benefits. So we did also animals with traumatic brain injury, looking at motor deficits in that case. The animal models of cognitive function, they don't read, they don't talk. It's sort of like, what would you do? They turn left, they turn right, but that's not quite the same thing. So we have done some simple tasks with animals, and we're very interested in pushing this forward. So vagus nerve stimulation for people with epilepsy, a lot of people got better in their mood without getting better at their epilepsy. And so it's actually FDA approved for depression as well. So we look for these ancillary benefits, and one obvious one is if someone had a stroke and the hand was like this, but they also had, let's say, some language problem or some memory problem. Could you then pair some kinds of cognitive training, adding, whether it's Brain HQ, one of Mike Merznick's tools from Posit Science or something else, or just reading our crossword puzzles. We don't know what's possible, but the general principles are the same. It doesn't really matter if you're moving your hand, which we think of as pretty simple, reach and grab a coin, but when you look at how complicated it is mechanically, a hand is crazy complicated. 27 degrees of freedom in each hand, that's a lot of ways to move the hand. So we don't think these cognitive tasks, even language, are out of reach for this technology to help. So you can imagine someone trying to do word finding, and they're trying to remember the word, or they're trying to complete a sentence or answer a question or do rhyming or whatever it might be. You could add vagus nerve stimulation when they successfully retrieve from their network that answer and push them toward better function. Haven't done any experiments on that yet. We have a speculative paper that we published. We did do experiments in the animals. They don't speak, but they do move their jaw. So we did at least show that you can enhance the number of motor cortex neurons that produce movements of the jaw, which is, again, that's what you can do in rats. We've also done a bunch of experiments in rats. They can't hear speech, but they can, they don't hear speech as speech, but they can hear all the speech sounds. So we have trained animals in all the English consonants and vowels, and we have shown that if you pair vagus nerve stimulation with a word like dad, you do enhance the brain's response to the sound D and not the sound S. So they're better at dad, not sad, for example. So you can get the same targeting. Again, that's just a sensory process, that's not language. But the principles seem to apply that you can use this technology to label stimuli and allow the brain to then enhance that and hopefully recover function.
Hi, what happens if you increase the frequency or amplitude in vagus nerve stimulation in a non-impaired human? Would you have someone who could learn and do things faster, like a superhuman?
That's a great question. So Dr. Krausik mentioned the Targeted Neuroplasticity Training Program, TNT, that the Defense Advanced Research Projects Agency funded, based on this general idea. They said, look, this is great. You ever heard from Admiral Craven, you know that most of what the military does is train people, because they all leave and they have train them again and again. They're really interested in could we enhance the rate of learning so that we could take our huge budget and train people faster and make less time and all the rest. I'm very surprised, somewhat surprised by the answer, which is no. And the reason for that is you're already really good at this. Evolution has given you 1 billion years of a head start on how to learn. Whether you're learning to move your arm, it doesn't matter if that arm is driving a bicycle or flying the space shuttle, you can figure it out. I can learn any human language, it doesn't really matter. I can do it at any age. It doesn't really matter. I may not be perfect at it, but I can do pretty good. Why are these people with serious mental health problems unable to get better? They don't have that ability. They have a genetic disorder, they have an environmental thing that was unanticipated. Why does someone who had a stroke or a spinal cord injury not able to get better? They don't have the luxury of an intact nervous system evaluation network that allows it to fix itself. And so the idea here is if we could restore it in those people who need help, then we would bring up the bottom without just advancing the top. So most technologies, as you know, the internet or anything else, performance enhancement helps everybody, but then you're still behind. It doesn't do you any good. This is one of those rare technologies, which is just a surprise, that it would help those who are impaired and doesn't help those who are blessed to have an intact brain. Good question.
Hi, excellent talk and very exciting. I was wondering if you see a difference in your results between right hemiparesis and left hemiparesis.
It's a great question. So now we have 150 people with hemiparesis, single-sided weakness. And so we can go back and do those studies. We were underpowered. If you only have 10, you can't really tell. With 150, you really ought to be able to tell. And we have looked at time since injury, right versus left, male and female, age, everything we've looked at, nothing predicts whether you make progress. That's two ways to look at that. One is, how can nothing explain anything? Well, you've looked at so much, so it happens that a clinical scan doesn't explain anything either. The size of the stroke doesn't actually predict how weak you'll be, which is, to me, surprising. Bigger strokes don't cause more problems. But that leads us to believe the brain's response to stroke is really the problem, or much of the problem. And so that means the neuroplasticity that was pathological, that was produced by the stroke, is causing much of the problem. And so the things you would obviously think would relate, has it been 10 years? Are you 5 years old? Age didn't matter, none of these things mattered. So we think of that as a glass half full. It's a little frustrating when some people get better and some people don't, and we don't know why. We would hope we could pick the people it would work better for and only implant them, and then the benefit would be bigger. We can't do that. We can't select the winners. We don't know who the winners are going to be. But we think it's By making adjustments to our technology, we'll be able to move people who didn't respond into response. But there are no predictors clinically of who will respond.
Okay, an online question. I'll actually combine two questions here, which I think are both relevant. With vagal nerve stimulation, the question is, does it sort of affect more synapses at the right window of time? Or is it an exaggerated or stronger response in a limited set of cells? That's kind of part one, a mechanism. And then secondly, are you applying this to aphasia?
Mm-hmm. So first question's a great one. This is Texas, and bigger is always better, and so you always want stronger. Who wants to be weaker? Nobody. So we always think of what we need to do is make more connections, if they were just more connections. We always think Einstein's smart because he had a big brain. None of that was true. You want the right connections, not more. And so what we see in the animals is it's just as important to weaken certain ones as to strengthen others. So we can inject, for example, opposing muscles. Now, if you want to make a rigid arm, you might want to simultaneously contract it. But if you've got a neuron in your brain that is connected to both flexion and extension, you're not going to do very well because you're going to activate them and you're going to get rigidity, which is what a lot of these people have. In the animals, we can literally see certain synapses weakening and other synapses strengthening so that what you lead to is a more healthy network the way that it's supposed to be. So that's the goal. Is it perfect? No, there's still disability. We're still working toward it. But it's not just about making it stronger. It's also about making the right ones weaker. And that makes the problem a lot harder. If it was just strengthening everything, that'd be easy. But that isn't how it works. You got to get just the right ones. On aphasia, our paper was mostly about apraxia of speech, which is the movement patterns of having trouble with speech. But even the higher cognitive, the language functions, there's not really a specific reason to believe that you couldn't also benefit from the same concepts. Speech is complicated, the hand is complicated. We don't know yet. There are a couple reasons to worry about it, why it might be a challenge. One is that when we activate the vagus nerve, we also activate all the other fibers that run through it. One of the largest ones, and one of the large ones, is the recurrent laryngeal nerve that goes from your brain to your larynx, runs through the vagus nerve. And so it might be that for people with vocal problems, you wouldn't want to be activating something that also activates the larynx. But it might be that that's actually a benefit. In many ways, we've been lucky that this bizarre combination of parasympathetic action and central action has worked out. And a lot of useful treatments end up being serendipitous. So we don't know yet whether that activation is helpful or harmful, and whether or not there really is an opportunity for aphasia or not. We'll have to do human clinical trials because, as I mentioned, there's not really an animal model of not being able to find the right word.
Okay, well, with that, let's thank Dr. Kilgard once again. It's an amazing journey. He's taking in what a remarkable program of research, and it's especially inspiring to get to hear some of the benefits that are happening for real people out in the world, but also the very careful, well thought out backstory behind what's driving this research forward. So we are all about neuroplasticity and lifelong brain health. We'll see you again on March 3rd when Dr. Alva Tang will talk about social and emotional development in the context of mental health. We'll see you then.